with the title Deep Learning-Based Input Function in Positron Emission Tomography Imaging. So the story of an innovation project. This project has evolved over the last 5 years maybe, and the innovation track has enabled us to get some funding for this project, which I will tell you more about. So just to give you a short introduction to positron emission tomography imaging, many of you have seen these slides before, but I'll go through them anyways. So the concept of PET imaging is that you have a radioactive atom that you somehow inject into the patient you wish to image. And when this decays, a positron is emitted and recombined with an electron, and then 2 gamma photons are emitted. And these are emitted in opposite directions. And this emission allows to be detected in a PET scanner. If 2 detectors in this ring are activated at the same time, you know that somewhere along this line there were an emission of these photons. So when you have millions of these events per second, you get somehow can reconstruct the distribution of the injected radioactive compound in the patient's body. So, for example, here you see a lung cancer where the tumor has received taken up a lot of this tracer. One of the key advantages with PET compared to other imaging modalities like MR and CT is that the units of these image voxels are becquerel per volume. So this is directly related to the physical event that happened, that occurred in the body. While in MR and CT, the units is more arbitrary. But becquerel is the inverse second, so it's actually the number of disintegrations per second. And this is very important for what I will tell you later. Yeah. To accomplish this, you have to apply a number of corrections. I will not go through them now. And also, another important concept in PET is that you have to have this radioactive atom injected into the patient. And there are several of these available that you see here. Some of them have very short half-life, 2 minutes, until half of it is decayed. But the most commonly used is this F18 with 2-hour half-life. And this is not enough. You also need to couple your radioactive atom onto a carrying molecule that allows you to image a certain biological process in the subject. Again, the most commonly used is the fluorodeoxyglucose, which is a glucose analog, but there are also other tracers. So you can, in the same patient, inject different tracers and get totally different images. And this is one of the advantages with PET. These 2 I will show you examples of later. So this is how it looks from a chemical point of view. So you need some chemists who do just replace one of the hydroxyl groups with this radioactive atom here. And then you have radio-labeled glucose in this case, which follows almost the same path as normal glucose, and you can do PET imaging with this. So I will come back to how this compartment model can help us in a while. Okay, we have the units of becquerel per volume. Then just to make it a bit simpler, we normalize these units to normalize away the injected dose and the body weight of the patient to minimize the variations within the population. Then we get the units of standardized uptake value, SUV. This is also a concept I will come back to. So in a static PET image, this is an average over the scan time. So maybe you put your patient in the scanner for 20 minutes, and then the image will be just an average of these 20 minutes. A more interesting concept from the quantification point of view is that you put your patient, or in this case a mouse, in the PET scanner, And you start the scan and start the injection at the same time of the tracer. Then you can follow the uptake of the tracer, of the radio tracer, over time. So here comes the tracer into the vena cava, and after some time it's distributed to different regions in the body. And then you can delineate regions and follow different organs, how they take up this compound. This is called dynamic PET. This is what I mostly do my research within. So what is the advantage with dynamic PET is that you can do this tracer kinetic modeling. And this is actually the gold standard way to quantify data in PET. So here is a conceptual image where you have your radio labeled tracer in the blood plasma. You inject the tracer in blood, and it's taken up into the free interstitium, where it's further taken up into cells or bound to receptors on the cell surface, and also eventually released and diffusing back into the blood. And these transports of the tracer are described by these rate constants, K1, K2, K3, and K4. And this process can be modeled in these compartment models, where you have arterial, the tracer in arterial blood, and the uptake or transfer into the different states of the tracer. So if you have the everything in the green box, this can be measured with PET. So if you have the tissue concentration of an organ or a region in the image, and if you also have the concentration of the tracer in the blood over time, then you can put up this model and solve the differential equations for this dynamic system, and you get the relation between these two with the rate constants involved. And if you have these two from measurements, you can fit the K values. And these K values are actually have the physical meaning of the transport of the tracer. So in the case of this glucose molecule, for example, Ki is proportional to the metabolic rate of glucose. So simply by injecting a meaningful tracer, you can measure a biological effect in the body of a patient non-invasively. So this is the great advantage with PET, which is not possible in other imaging modalities. So one of the problems with this tracer kinetic modeling is that you need this input function. And especially in small mice, but also in humans, this is a time-consuming procedure. In mice, it's also terminal. You cannot do longitudinal experiments because you terminate the mouse afterwards, and there's some limited blood volume. So people do simplifications. They find some artery in the image, and they delineate, make a region, a segmentation of this, and extract the input function from this region. This also has to apply some corrections onto these with this method. So this is not far from ideal. So this is where our approach comes in. So what we try to do is to predict this input function, this curve from the images directly. So we call this deep learning derived input functions, or DLIF. Some of the work that led up to this, if I call it invention, was things I did during the PhD a few years ago. Here we had a slightly different approach. So we delineated some regions in the mice, in this case, in the PET images, extracted the uptake curves, the time activity curves of, say, 5 or 6 regions, and we trained the machine learning model to basically transform these 5 curves into the arterial input function. So we had one publication showing that this was possible in mice, and also another paper on where we tried this in humans in an oxygen, it should be 15 water study. So we saw that the concept sort of worked, that you could transform these image-derived curves into a useful input function. However, someone then has to go in and delineate these regions, so it's still a human interaction. So we wanted to overcome this manual interaction with the images. So we moved on, tried to develop this further and see if we could input the images directly. So these are 4D hypercubes, 3D volumes over time. If we could input these into a neural network and perform spatial and temporal feature extraction and then regression into this useful arterial input function curve. So we had some retrospective training data to do this, collected at UIT many years ago. Unfortunately, these training data did not have blood sampling as ground truth, but only an image-derived input function as ground truth. So it was not so easy to convince the reviewers that this was promising. So we found some collaborators in Canada and got a few mice where we got blood sampling. So we used these for testing. We trained the model on the image-derived input function and tested on real blood sample mice. And this is what so far led up to the work we've done so far. Here is the small animal PET scanner, by the way. And this is a mouse PET image. You see the heart and the kidneys and the bladder. And the results looked quite promising. The red is the ground truth blood for one of the 13 test mice where we had real blood samples. And the green is the average and standard deviation of the deep learning based input function models, predictions. So there was a promising agreement, and we could move on and perform this tissue compartment modeling and get this, if you remember, the K values, which are the measurements you actually get from PET. And we saw that there was a nice correlation between the K values derived with a blood input function and the K values derived from the deep learning based input function. And this is in two different tissues, in myocardium, which is the heart, and in tumor, because these mice had an implanted tumor. So okay, this was promising, and we were looking forward to move on. So what we did was that we, in December 21, submitted what is called DOFI. Now I forgot the word, the Declaration of Invention. And actually we learned that if you work on a problem at your university and you have an idea that potentially has commercial impact, you're actually obliged to submit a DOFI to your university because they own the potential invention. So we did this and started working together with our technology transfer office, Nordinova, and they applied for this qualification project and got the money from NRC. So we visited some conferences with this idea, talked to the industry, and got a feeling about if there was a market for this idea. And we were also granted a 2-year position from our university to go ahead to collect more data and to establish this methodology here at UIT to collect new real training data. Then we also had 2 master students working on this. I'll show some results of their work in a while. And now we're applying for the next step, which is called the verification project from the Research Council, which is a 5 million and 2-year track where you should bring your product to the market. I had some oral presentation in some conferences, and very soon we will start writing some papers that wrap up these ideas. So just to tell you a bit more about this 2-year grant that we got. So we got money to hire a biologist in our group to collect more training data. So basically to develop this idea into a working model. And one of the things we started with was to investigate how different variables affected the shape of the input function, because we wanted to know how robust, how similar must the training data be to train models, and we needed to validate this properly. And by the way, here's a promotional image of the team. You might recognize some faces here. This is Christophe and Luigi from our team, Rodrigo and myself. And this is the preclinical PET MR scanner. Yeah, so I will quickly just go through the methodology, because this is, from a biological point of view, quite astonishing that Rodrigo, my colleague, can manage to do these surgeries, because the artery in the mice are very, very small. So what we do is we isolate the artery and we make this loop and loop the blood through a detector to measure the true input function. And we do this while we do the PET scan. So to learn this, we had to go to University of Sherbrooke in Canada, because this group has this as a routine in their lab. Very good visit in September last year. And this is how it looks when we do the surgeries here. So here's a mouse. First, we do a tail vein injection, a tail vein catheterization. So you have to try to stinch the vein in the tail with a catheter like this. This is quite standard in the mouse research to do this. However, to do the arterial cannulation, arteries are much deeper and much smaller than veins. So you need to dissect out the artery and find the artery. Here it is. And then make a small incision with a very sharp scissor, and with a small polyethylene tube, insert it in the artery, and then tie it so that it's fixated. And then you can hook on the arterial line into this coincidence probe, further to a pump, and then through a Y connection back in the vein. So we have this loop, and the pump makes it circulating constantly. And this is very important because you cannot extract that much blood from a mouse. So we need to put it back again. And here you see live how it looks. The mouse is in the PET scanner, and then this is the detector and the pump, and the blood is circulating. And here is the Y connection where we inject the tracer while the blood is circulating. And this is an example of a input function, of a good quality input function measured in our lab. We also take some blood samples to calibrate the curve and etc. Yeah, very quickly we have currently scanned around 60 successful mice, but more than double because of all the failures and the training that we needed to do to establish the methodology. We are currently collecting the last mice for this validation study, where we check different injection volumes, injection time, withdrawal rate, and mouse age and mouse strain. So, because if we want to train these models, we have to know if the input will vary and if the output will vary with these variables. If we don't know this, we don't know what's causing the variations in the models. So just to quickly show you some of the results, these are, for example, if we inject the tracer during 15 seconds, during 30 seconds, or during 60 seconds time. So if I show you all the curves, it looks like this, but if I show you the mean and the ±1 standard deviation, it looks like this. So evidently, I have it bigger here, the short injections give a sharper peak, and very slow injections, 1 minute, gives a more spread out peak. And this is important because if the model gets an input data which does not reflect this, but the ground truth output reflect this change, the model will never be able to learn this behavior of the ground truth. So we need to make sure that the model has the possibility to learn this. Also, different withdrawal rates on the pump has an effect. The higher the withdrawal rate, the sharper the peak. While the injection volume and the mouse age does not seem to affect the results. Also, we did something to inject 3 times in the same mouse during one single scan, and we will later overlay these 3 to check how really reproducible the input function is. Yeah, also I've not done it yet, but we will of course compare these curves statistically to make sure that it's what is significant and not. So we will extract the features from these curves and check that if they are really different or not. And then we will move on to delineate regions in the mice and do the compartment modeling with both the input function and the image-derived input function as input, and compare the KI values. And then later we can use this data to train our deep learning models. So that was like the data collection part. But we also done some deep learning work on the retrospective data. And this was actually presented by our master student, Fredrik Aspheim, yesterday in a conference in Bergen. The deep learning models that, if you remember the slide where I showed, where you have the transformation from the images down to the input function. We started out with models like ResNet or other architectures, where the limitation of these were that we have some fully connected layers in the end, and this will limit the network to a fixed number of time steps, for example. However, the results of these early models were quite promising. As you see, the ground truth and the predictions were okay. But we still needed to develop a method to understand and interpret these predictions. So one way to do this is to use attention maps, which has to be baked into the models during training. And then you can see what the model will focus on. Another idea is to use salience maps, which are more used in classification tasks. So it's not straightforward how to do this for regression in this case. And the third option is to use masking in the input during testing to remove information and see how the output changes. So when we did this, this is the masking approach for these early networks. Here is the original image. And when we put a threshold on the image voxels, we saw that the output did not change. So these 2 curves still look the same, region and thresholded prediction. This means that the model did not learn actually anything from the input data. So what did the model learn then? Well, this was a classical example of a shortcut learning. The model just learned the average data, actually. So what we discovered was that in the convolutional layers and in the fully connected layers, there was this bias term that basically overtook the predictions. So all the weights were 0, but the bias learned everything. So it learned the average curve. So what we did, we had to remove all bias terms, and we also introduced attention into the networks. So the architecture that we're currently working on, and it's not entirely finished yet, and I cannot disclose more details than this, but it has some attention gates where we actually can go in after training and look what the model focus on. And it also has, so it works in a similar way. It extracts 3D features from each time step in the first step, and then we make them into a sequence of time, and then perform temporal extraction of features using 4D convolutions. And then another difference we also did now in the last weeks was that we introduced fully convolutional layers in the end, down to one feature, so that we can use any number of time steps as output. This gives a more flexible network. And when we did this simple thresholding test with the new networks, we actually saw that when we masked away different thresholds, the result changed. So obviously, if you remove these high intensity voxels, the peak is underestimated in the dashed line. And if we remove even more, the peak is even more underestimated, also the tail here. So now the network is actually learning some meaningful information from the input, not just the bias that learned the average in the training data. So this is promising. We're still working on fine-tuning all the details in the networks, and we will also go on look into the attention maps. Here we haven't, this is really recent, we haven't had time to look into this, but it seems like there is some focus in the early time steps here in the vena cava, where the tracer comes in, and in the late time steps here in the kidneys, where the tracer is taken up in the late time frames. So this makes sense from a biological point of view as well. Yeah, and this kinetic modeling on these latest results is also promising. And we cannot wait to apply these models to the new training data where we have ground truth blood data. So to conclude, we have established arterial blood sampling with simultaneous PET at UIT. We saw that short times, injection times are preferable and highest possible withdrawal rate. So in the new training data, we will use this knowledge. And we're working on the models, new architectures that generalize well and that allows interpretability. So hopefully in the future, this knowledge can allow us to evade blood sampling in mice to do longitudinal studies, but it can also later be transferred to humans and allow dynamic PET to become part of the clinical workflow, which is it's not currently done so much. So with this, I would just like to make a final announcement. I'm also part of a committee arranging this autumn school for research in AI in medical imaging. This was arranged last year in Bergen in September, and this year we are arranging it in Sommarøya outside Tromsø. So 13th to 15th of September. You're welcome to join PhD candidates, postdocs, master students, clinical researchers. It's almost free, just 500 crowns. Presimal covers the rest. So it will be a 3-day workshop. So registration open today. So hurry to register and you get a place. It's first come, first served. So with this, I would like to thanks for your attention.